Hello, my name is Cameron, and today we're going to be looking at cell division, specifically mitosis, stem cells, and cloning. So what is mitosis? Mitosis is going to be the replication of cells. Cells are going to split or divide into two genetically identical daughter cells that are the same as the original cell, meaning they are going to contain the same number of chromosomes. Okay, so this happens in every multicellular eukaryotic organism, okay, except for our gametes or our sex cells. They go through meiosis, but all other cells of your body go through mitosis to replicate. Now, what's the purpose of this? Is to help the organism grow, to help them repair themselves, okay, and to just generally develop, okay. So, as we said, the product of mitosis is going to be two genetically identical daughter cells containing a full set of chromosomes. We represent that as 2n. So there are 4 stages of mitosis that you will have to learn on your level. It starts out with the cell that has the full number of chromosomes, 46. Okay, so this is going to be made up of 2 sets of chromosomes, 23 each. Each set is going to be from your parents, combining to form a full set, which is going to be you. Stage 2, the cells are going to replicate their DNA, and they're now going to contain a total amount of 92 chromosomes. So 2 sets of the full number of chromosomes. They do this because in stage 3, the cell is going to divide and separate into 2 cells now containing both 46 chromosomes, or the full set of chromosomes. Okay, they do this by lining up along the equator of the cell, and then they are split using spindle fibers. They're going to attach to the centromeres of the chromosomes and pull them apart. So now you're going to have stage 4, the cell cytoplasm is going to split, called cytokinesis, and this is where those 46 chromosomes on either side of the cell are now going to become part of 2 genetically identical daughter cells. This process will then carry on as these cells grow and replicate again and again. So something I always mention when it comes to biology: have a vocabulary list. Have a list of all the difficult terms that you are going to come across in that topic. It is very difficult to remember all of these terms, so you just have to constantly use them in giving full answers that contain the words of that topic. In this topic, we're looking at the words chromosome, gene, and DNA. So it starts out with DNA. Okay, a DNA is a double helix structure made of all your genes. Your genes are something that gives a specific trait to you through the use of proteins. So DNA is made up of sections called genes. These genes are going to encode for a certain protein. That protein is going to have a specific function in your body, giving you a certain trait. Okay. Now you have an incredible amount of DNA, and that all has to be packaged into these packages. Okay, and these packages, we call them chromosomes. So these are small sections of your DNA. You have 46 chromosomes, and they make up our genome. Okay, and they're also found in pairs. I like to say that there are chromatids. Okay, 2 chromatids are going to form 1 chromosome. Okay, so we've come across the word stem cells. Now, what are stem cells? Stem cells are undifferentiated cells that have not been specialized and don't have a specific function. In the previous section, we talked about specialized cells are cells that are adapted to perform a certain function, like your nerve cells, your muscle cells. But stem cells are cells that could become anything. Now, when you start out as an embryo, okay, the sperm and the egg have fertilized one another, and it starts to grow. It is just replicating. It hasn't figured out exactly what it needs to make up that organism just yet. So those are called stem cells. They're undifferentiated, unspecialized. They are just a base cell. Okay. And they can specialize to form any cell that you may need. Now, why is this important? Because we can use stem cells, okay, in research and development of medicines, in the growth of possibly organs one day for organ transplant patients. And we get into this a bit more in cloning in later sections and a bit in the next slide. Okay, so they can also be seen as unethical because, of course, at what point does it become a person? At what point do those little clumps of cells that form a fetus, become a living person? Okay, so if you're taking out those cells, are you taking out a person? These are the ethics that come with medicine and genetics. Okay, and it's also against a lot of religions and faiths to do this sort of practice. At the same time, it can save a lot of lives, because imagine that you need a heart transplant, and instead of waiting for someone to die, they grow you your new heart. Let's say that there's a new treatment that might save your life or improve your life drastically. They can test it on some of your cells first before giving it to you, therefore making sure you don't have any adverse reactions to the medicine that might kill you if you weren't able to be tested on beforehand. Okay, again, when it comes to cloning, you can either use it for tissue cloning in therapeutic cloning. Okay, this is like growing skin grafts, growing new healthy cells that can help your organs rejuvenate. Okay, it can replace damaged cells or cells that are inactive, such as cells in your pancreas to produce insulin. When it comes to cloning, you can also use it in plants. Okay, if there is a specific variety that you want, you can clone that plant by taking a group of these stem cells from the plant and propagating them, growing them in a dish to make many other cells or other plants from that one sample. Whereas usually you would have to wait for the seeds to be fertilized and the seeds to grow. Okay, and then in human and animal cloning, the same thing. It is going to be using these stem cells, and you can make a genetically identical copy of an animal, let's say. Okay, so you don't have to wait for a heifer or a female cow to be pregnant for a long stage of time before having another child. If you want those certain genetics, you can just artificially inseminate these cloned cells into many different cows, and we get more into that in later lectures. So, my name is Cameron, and this has been cellular division. I'll see you in the next lesson.